I hope you are still focused after this long but interesting day. And I will like to give you some overview about the strigolactones and my recent research about them. My research was conducted in the cooperation with the Agata Daszkowska-Golec and the Marek Marzec. So I would like to start with the introduction with some words about the strigolactones. So strigolactones are the youngest class of phytohormones, which synthesis takes place in roots from where they're transported to the above ground part of the plant. And today, about more than 30 naturally occurring strigolactones have been identified among the monocots and the dicot species, which they can play many roles in the plant growth and development. For example, there are the major role of the strigolactones are the controlling the architecture of the plant by inhibiting the branching of the shoots and the roots. However, also their participation was proved in the seed germination, the early seedling development, senescence, fruit hair elongation. And more research also proved their participation in the plant's response to the abiotic stress, such as solid salinity or the drought. And in our lab, using the tilling strategy, we was able to identify the mutant D14.D, which caused the missense mutation in the gene encoding the strigolactone specific receptor, the D14 protein. And plants harboring this mutation in the D14 gene are insensitive to strigolactone treatment and produce almost twice as many killers as the white-eyed plants, the genotype Sebastian. And also the mutants possess shorter but more branched roots when compared to the white-eyed. So as the strigolactones are the newest class of phytohormone, they were included to the phytohormones class in the 2008. No transcription factor has been identified that could control the response of barley to the strigolactone. So here, using the strigolactone insensitive mutant, the D14.D, we were able to select the transcription factors that may be involved in the regulating expression of strigolactone dependent genes in the barley. So moving to the methods, the first step was the RNA sequencing analysis of the shoot tissue to select the strigolactone responsive genes in barley. And the tissue dedicated to the RNA isolation was collected from the 2 weeks old seedlings of the white-eyed and of course the strigolactone insensitive mutant. And both these genotypes were treated with the 2 concentration of the strigolactone synthetic analog, which is the 1 micromole of the strigolactone and 10 micromoles of the strigolactone. And after the half hour and 3 hours of the treatment, the tissue was collected to isolate the RNA. And of course, the control plants were sprayed with the equimolar concentration of the acetone. Acetone, because of the powder strigolactone is dissolved in the acetone. And to focus on the strigolactone-related genes, we consider a gene to be differentially expressed when its quotient was higher, above 1 or lower than -1 for the down and up related genes respectively. Yeah, so you can hear me, yeah?
Yes, we can hear you.
Because I hear something stop here, okay. So moving forward to the results, using the before mentioned criteria, we identified in total 139 differentially expressed genes in the white type and 27 differentially expressed genes in the mutant. And as you can see, it is easy to observe that we can observe that not a lot of genes were upregulated or downregulated after the stragolactone treatment in mutant, which proved that the mutant is insensitive to the stragolactone. And after the comparison of the differentially expressed genes between the wild type and the mutant, we can observe that no common genes or a single common genes, of course, corresponding time and the stragolactone concentration, were common for these two genotypes. So overall, summing up the genes which were altered with the stragolactone treatment in the wild type, which is 79, 23, 30, and the 15, after the removing the duplicated genes, we were able to detect over 100 of stragolactone specific genes. Yes, so moving forward, then we use the Biomalt tool, which is available on the Ensemble Plants platform. We're able to download the promoter sequences, which is the 1,500 before the codon start of the 92 stragolactone responsive genes which were previously detected. Next, using the PlantRec map tool, also available online tool, the potentially regulatory interaction between the transcription factors and the input genes were identified. And further, we were able to detect the 47 transcription factors with the overrepresented targets in the input genes. And these 37 transcriptor factors may be considered to be involved in the stragolactone signaling transduction pathway. As you can see, I select 6 of them. 6 of these transcriptor factors control more than half or almost half of the genes whose expression was altered by the treatment. You can see it here. It's about 30, 34, 40, 60, 38, and 31 genes, up to 92 genes which were as an input. You can see here, this is the block ID of the barley transcription factor, and also here, this is the homologue in the Arabidopsis. And the detected transcription factors are the ethylene responsive element binding factor 5, the basic pentacysteine 6, the baby boom transcription factor, and the dehydration responsive element binding factor, and the cytokinin response factor 4. And they participate in the in the organ morphogenesis, like for example baby boom and the leaf development. However, also they it's supposed that stragolactone may interact with the other hormones, because we can see that there's an ethylene responsive binding factor or the cytokinin responsive factor, and also to the draft response. As I said before, that the stragolactones are strongly believed to also participate in the plant response to the abiotic stress. And here in the picture, there's a sequences of the binding parts of these transcription factors, and this may be considered as a regulatory element, stragolactone regulatory elements, which may be conserved both in the dicots plants and in monocots plants, because they are both in the barley and the Arabidopsis transcription factors. And moving to the conclusions, the analysis revealed that we will reveal the set of transcription factors that may be involved in the stragolactone signaling pathway in barley, and also our analysis proved that our mutant, in which were created in our lab, is the stragolactone insensitive mutant, and which is not able to respond to the stragolactone. Yes. That's all what I wanted to say about my research, and that's all. Thank you.